in Planta transformation. So this is another technique where we can minimize the use of plant tissue culture technique, या दूसरे अल्फाज में कहा जाए कि मिनिमाइज करने के अलावा जो टिश्यू कल्चर की रिक्वायरमेंट है इसको ऑल टुगेदर रिप्लेस कर दिया गया है in this case of in planta transformation. Now, in this technique, the growing plant, it can be transformed. That is, in this case, one can say that some plant cells, explants, their use, it can be minimized. And this transformation, it can be carried out at an appropriate stage of the life cycle of the plant. जो प्लांट इसमें यूज़ किया गया है वो एक मॉडल प्लांट है. द नेम इज़ वेरी कॉमन, दैट इज़ अरेबिडोप्सिस थैलियाना. ये एक मॉडल प्लांट की टाइप है और इसको बहुत सारे एक्सपेरिमेंटेशन में यूज़ किया गया है, लाइक इन सेल बायोलॉजी, मोलेकुलर बायोलॉजी, even to study a variety of plant microbe interaction experiments. Reason is that its genome comparatively it is smaller as compared to other higher plants, and its lifespan it is short, so that within a few weeks we can get the mature plant. So for this purpose, this plant it is highly desirable to perform some molecular biology techniques. और इसमें जो सीड्स बनते हैं it is around 1000 rather 10,000 per plants. So as I told you that at an appropriate stage of the life cycle it can be exploited to transform this whole plant. For example, one can say that when pollen grains they are going to mature or at the stage of gamete formation, explants or rather than apical parts like floral parts, they can be transformed. And here a system that may be based on agrobacterium tumefacients, they can be used. So most of the study have been carried out by using this model plant Arabidopsis thaliana, but the success with other plants it is limited. So because of this reason, in Planta transformation, it has not been exploited in much detail. So hopefully now you are well aware what it is meant by in Planta transformation, that here transformation it is carried out with intact plant during its life cycle. So in this case, variety of techniques have been used. So one of the technique is that to transform floral buds, that when plants they are growing at the flowering stage, then they can be incubated with the suspension of agrobacterium, and then they can be further selected for transformation. So such a type of techniques has been reported. In addition to that, with the utilization of minor tissue culture technique, some transformation in Planta has been carried out. For example, growing meristem parts, meristems they are actively dividing parts of the plants that are mainly present at the apices, at the apex of shoot or root. They can be transformed, or they can be even transformed with agrobacterium tumefacients, or some particle bombardment can be used for transformation. And then after transformation, transformed cells they can be further selected by using some minor tissue culture techniques. So that's why I told you initially that it has minimized the use of plant tissue culture techniques. Otherwise, some techniques can be carried out where we can transform intact plants, and the most common example is Arabidopsis thaliana.